Welcome all. This is my presentation on Pathline, a tool for comparative functional genomics. Let's begin. We're going to start by summarizing the content of the article. So let's talk about the types of data that Pathline was developed for. This is all in the field of functional genomics, and there are three key types of data in this field. Functional data, which is gene activity measurements. Pathway data, which is the series of reactions within a cellular process, and phylogenetic relationship data, which is the relatedness of species over time. Researchers need to accomplish tasks at three distinct levels. There's the detailed comparison of a limited number of time series. There's an aggregate comparison of similarity scores across a pathway, and multiple similarity score comparison. In response to these pressures, the authors of this paper have created Pathline. Pathline is a new interactive tool, and it facilitates research across all three data types and all three levels, and it does so in a very charming visual interface. The creators of Pathline believe that it will be useful for a variety of phylogenetic data. However, they did have a specific data set in mind when they created the tool, and it was used as their example when they wrote the paper, and it's pretty much the only data set we'll be talking about for the rest of this presentation. So this data set is a collection of pathways that make up the metabolism in yeast. Each pathway consists of specialized gene products that process chemical compounds called metabolites. The data is similar across all the species of yeast, but it is different in each species, and it may be different in a number of ways. A gene, for example, might turn on earlier in one species than it does in another. Some species have a different control mechanism for a gene. Some species have, of course, evolved novel genes altogether. Pathline was designed with the goal of looking at multiple sets of values simultaneously across time, species, genes, and metabolites in order to find patterns across all those variables. The design of Pathline includes two linked components. Our book would refer to them as facets. These components are original designs created by the authors in order to meet the goal of their end users. The first visual design component we're going to discuss is the curve map. On the right, you can see a snapshot of the Pathline interface, and the curve map is this component that takes up the majority of the interface. Each row of the curve map represents a different species, in our case, a species of yeast. The order of the rows is determined by their relatedness, as shown in this phylogenetic tree that's along the left side of the visual. The columns, meanwhile, show gene or metabolite measurement. They are in the order that the user selects them. Along the bottom and right of the visual are overlay plots. The bottom overlay represents all the curves for a given gene expression or metabolite, and the right overlay represents all the curves for a given species of yeast. The other visual component is called linearized pathways. This component, which is on the left of the interface, shows similarity scores for genes/metabolites encoded with horizontal spatial positioning. You can view up to 3 similarity scores at a time, and the process by which this visual is made is a lot more complex and possibly more interesting than the curve map. In order to make the linearized pathways, you start with a very different visual. So here we have the process of a metabolism where each node or point is a metabolite, each line is a gene, and There are branch points that are easily visible. There are cycles that are very clearly visible. But that doesn't translate very well into the almost 1D representation that is desirable. So we unwind cycles, we linearize the process, we take branches and set them aside, and remap the entire design so that it's all linear and straight. We end up with this final visual where we still are able to show cycles because we show where the cycle starts with this glyph. We're able to show branches with these glyphs, and that way we can represent the entire metabolite cycle here along the side of this visual where now the genes are points and the metabolites are lines, and the similarity of the gene is coded by its location in this space. In addition to that, aspects of the genes are color-coded. So if a gene is going in reverse, then it's blue. If a gene is going forward, it's green. And if a gene is bidirectional, it is orange. So hopefully that explanation worked for everyone, and you now have a decent idea about how Pathline is intended to function. The authors of the paper, for their part, were always thinking about how Pathline would be used in the field, and 7 biologists with years and years of experience between them were engaged from the very start in the design of Pathline. The process took 3 months of discussion, 2 months of refining prototypes, and the final product does seem to have been very successful. It's now the primary visualization tool for the team of biologists, and it's facilitated them in a variety of accomplishments, which they go into detail about in the paper. They were able to identify mistakes in pre-processing. They were able to dive deep into metabolites M18 and M19 for comparison, really showing how this tool can work at a variety of scales. They were able to discover that bidirectional enzymes share similar patterns across all species of yeast. The paper has a lot more examples than this that I don't have time to get into here, but it's clear that the team really valued this tool and found Pathline useful in just a variety of ways. So now I'm going to provide my own critique of Pathline and tell you what I thought of its design choices. So if you're looking at the images in this presentation, you might think that they look rather familiar, kind of like the images in our book. And I think that's very much by choice. The authors took a lot of care in justifying their design choices and referencing best practices when they were choosing to use spatial encoding for variables like similarity and choosing to use color encoding for their categories. They were always sure to explain why that was the best choice for their use case. I was unable to find anywhere really that the designers deviated from the advice in our text, and I think that the results speak for themselves, and I think it's a very lovely design. As we said at the start, Pathline wasn't just designed for the yeast metabolism use case, but it was somewhat limited exactly what the paper covered. Transferability was discussed, but it wasn't really experimented with in the paper. And because you're limited by the screen real estate and because of how complex the visuals are, I can imagine that there would be a fair number of data sets that it would be hard to apply Pathline to. If we could just talk about the components for a moment, I think that the grid component is really well designed. I like the idea. It's elegant to look at, it's artful. I think the information density is good, and it is very information dense. I would like to see a few more interaction tools. I believe that there will be a presentation today with a kind of similar use case visual that has more. It's also possible that different visuals could be more useful for different aspects of the data, and I wouldn't mind seeing that incorporated as well, maybe still in the same grid. I also really like the linearization view. It's hard for me to understand because I'm not a biologist and I don't have as strong a biology background as some of the people in this class, or certainly the designers, did. But I'm still super impressed. I haven't seen many visualizations like this before. It's pretty unique, and what it's trying to do and how it does it, it combines all these types of encoding, and it looks very pleasing and it's very compact. It's a shame that you can only see 3 similarity scores at a time, and also because it's so complicated, I imagine it might be hard to extend to different data sets in the future. The last thing I want to talk about is whether or not Pathline accomplishes all the goals that were set out at the beginning of this presentation. And I think it does. I think all types of data are well utilized. Also, I think all of the tasks are more or less accomplished. The curve map grid accomplishes the detailed comparison effectively. The aggregate curves should accomplish the aggregate comparison task. I think this could have been done possibly a little bit better if we had different plots, but that would add more to the user's mental map. It would add more to the interface. There'd be more to navigate. I think they did a really great job. And of course, the linearized pathways allow for multiple similarity score comparison, fulfilling that task as well. Although again, only 3 at a time. And that pretty much concludes my presentation on Pathline, a tool for comparative functional genomics. Here is my reference section, and now I will open up the floor or virtual floor to questions.